And I was asked to focus on novel methods. And so this was one of the options that the organizers chose for me to talk about because we are, in fact, using optogenetics pretty extensively. But the one thing that I would like to emphasize is that, especially for younger people, is that methods should not be used for the sake of methods. Methods should always be used they should be always at the service of scientific questions. So in that sense, one of the main scientific questions that I have had all my life is, why do we love dopamine so much? Dopamine cells are only, oh great, this works this way. Dopamine cells are small number of cells, less than 0.001% of the brain cells. They project to not a lot of places, frontal cortical areas and basal ganglia, especially dopamine cells in the ventral tegmental area, pretty much only go to ventral striatum and prefrontal cortex. A little bit of a projection to hippocampus, but not much. But we tend to put a huge amount of attention on dopamine. And it's not just scientists. The media loves it too. Dopamine is the only neurotransmitter with its own movie. I'm not making this up. There was a horrible movie that actually was made a few years ago. Initially, when we heard about this movie being made, those of us who do dopamine research were really excited because we thought that this is us, we're going to be famous till we saw the movie. But the reason I actually show this slide is look at the main line of advertisement for the movie: dopamine, chemistry or chemical. And this essentially sums up why we really like dopamine, because we think it's about chemistry and more. So when you think about it, you actually see dopamine being very much out in the pop culture because it is considered a reward chemical. This is actually an article that was in the Slate magazine. It's good entertainment. I recommend that you read it. But it goes on and on about whether dopamine is love, adultery, motivation, gambling, addiction, and the conclusion is that it is, in fact, all of the above. So with this in mind, the problem has been that this is entirely driven by the fact that dopamine is associated with reward. It signals reward. And so a lot of literature has been focused on this aspect of dopamine. But dopamine does a lot of other far more interesting things. So it's pretty much associated with just about any important cognitive high-order function. It's very much involved in attention, goal-directed movement, motivation, and affective processing. It's involved in multiple forms of learning and memory, and, of course, very much involved in executive functions such as working memory and decision-making. These are really, really interesting and important brain functions. And the reason we know that dopamine is involved is, of course, if you manipulate the dopamine system in any way, and that includes in humans with dopaminergic drugs, a whole host of these very important functions are affected. Dopamine is also intimately involved in brain disorders. And these include, are not limited to, ADHD, schizophrenia, addiction, mood disorders, and of course, Parkinson's disease and other movement disorders. These are a very complex group of disorders, and there's far more about these than reward. Okay, so how does dopamine do all this? So this is one of the questions that I have always had, and we have hardly been able to answer it. And we are beginning to, because of a lot of the cool tools that are at our possession now. So my lab primarily studies neural networks that maintain cognitive and emotional functions and that are relevant to psychiatric disorders. Up to about 5 to 10 years ago, we could record from a cell here and there and measure dopamine here and there, but now there's so many toys that are at our possession. So we are recording large number of cells in behaving animals, multiple brain regions, and readily applying optogenetics and other methods to manipulate this. Okay, so at the service of the dopamine function, how are we doing in the context of answering how dopamine does all the interesting stuff that it does? So if you go back and look at the reward circuitry, dopamine neurons respond really, really fast. They show millisecond bursts to sensory stimuli or learn responses to reward-related cues. This is a very classic response of the dopamine cell, and I'm actually going to show you some examples from my lab. We record from VTA cells. We have multi-electrodes in the ventral tegmental area, and we can detect both dopamine-like cells and GABA cells. And if you go and have the rat do a task that pairs a cue to an appetitive outcome, like a sugar pellet, or a cue to an aversive outcome, like a shock, you can see very nice, fast responses that change in the dopamine response. So here is, for example, this is cue, reward, session 1 of training, session 8 of training, and you can see this very fast few millisecond phasic response that from session 1 to session 8 increases in response to cue. And these are classic cue-reward association responses of to dopamine cells. Okay. And you can actually do far more interesting things. This signal is very flexible, so you can actually, one of the tasks that we have done, this is the work of Youngbuk Kim, by the way, 2 or 3 really nice papers. And you can pair one cue with something that's appetitive, food delivery, another one with something that's aversive, and present randomly these cue outcomes in the same training session, in the same learning session, so that you can track the same neurons. And you see very interesting changes in whether the animal associates a cue to a reward or a cue to an aversive outcome. So for example, this is during training session 5 and 6, this is light, which the animal has learned predicts reward. And the same animal has learned that tone is a shock. So you see that when it hears when it sees the light, there's a nice phasic response. Again, very fast, a few milliseconds, and not much. In the same animals, you can reverse the associations. Now you can pair the light to shock and tone to food, and you see beautiful shifting. So you see this phasic response to cue, how it goes away, whereas the response to tone, which was not much, when the animal learns that, oh, there's food involved here, that gets stronger. So this is something we've known about the dopamine cells, and I think it's really cool, it's really interesting, and they're very flexible in the way they respond to these changes in the environment, and they're encoding this reward expectancy, something that may happen in the future really nicely. But let's go back to these functions that we know dopamine does, these very interesting high-order functions. Let's think about these things. Let's think about your paying attention, or feeling really happy or unhappy, being motivated, working memory, when you have to make a decision. These constructs are slow constructs. You don't pay attention for a millisecond. Sustained attention, if it's at the service of a goal-directed behavior for you, has to last several seconds, several minutes. Motivation, emotions, those constructs, they last for minutes to hours. So how is it that dopamine cells are firing quick bursts only to an expected outcome, but yet dopamine is involved in these functions? So this is essentially what we've been trying to think about. So we know that dopamine does really, really fast stuff, fires really fast to cues, that if the cue is associated with something good in the future. But there's also literature suggesting that dopamine has a slow function, that there's slow changes about dopamine. And this is really interesting, and I've been really fascinated by this slow action of dopamine. And I'll show you some examples, but this is not trivial. We can't just not pay attention to it. Why? Because I don't care how fast a neuron fires. What really matters is the neurotransmitter being released downstream and then acting on postsynaptic receptors. That is what dictates neurotransmission. And with that, with dopamine, The postsynaptic cells are mostly quite distal from the site of release, especially in the prefrontal cortex, where a lot of receptors are actually up to a whole millimeter away from the site of release. So this is something that's really, really important. So if you actually, just an example of this slow mode of dopamine, if you measure dopamine influx in terminal areas, such as the prefrontal cortex, while an animal does a cognitive task, what happens? Well, dopamine levels stay up for a very long time. So an example I'm showing you here is an animal that's doing a cognitive flexibility set shifting task. I'm not going to go through the detail. If you're interested, this was a work of Mark Stefani. He was a postdoc in our lab about a decade ago. He was a master of designing wonderful maze tasks, so he designed this task. And essentially, the animal had many choices between color, texture, and turn, and the strategy would be very much like a Wisconsin karts test would change. So we would do 2 or 3 sets of strategy shifting in a given day while we're measuring dopamine. So if you look at one of these sessions in about 7 animals, this is set 1. Animal is figuring out what the rule is. So it figures it out within 20 minutes. And then an hour later, we put the animal back in the maze. It does set 2. That's to figure out that the rule has changed and shift. Okay, so dopamine goes up as the animal is in the maze trying to figure this out. And this is expected. But then after this 20 minutes maze period, we take the animal out and we put the animal in the home cage. So dopamine, as you can see, is staying up for 20, 40 minutes. The same happens later. So this dopamine that's staying up, what is it doing? Is it significant? So those are the sort of questions that we were trying to answer. And this sort of elevation has generally been dismissed as, oh, well, dopamine cells were bursting, a lot of dopamine was released, and it just hasn't been cleared by dopamine transporters. So it really is not important at that point because it's just leftover dopamine. Well, I don't think it's that simple, and that's something that we have been trying to figure out. And so essentially one of the things that we have been trying to answer is this fast response connected into the slow response? So the 2 questions then that we're trying to answer is A, how is this fast response to anything, environmental cue, just any event, related to these high order functions that I outlined for you? And how is this fast spiking related to sustained activation of dopamine release? Okay. So this is where I'm going to switch to 2 data sets. They're completely unpublished. Hopefully you'll see them published sooner than later, but they're both being written up and submitted, and both of them are a mix of cool new techniques and very old-fashioned scientific thinking. So the first one is, let's just tell you about the high-order functions. So if you think about all those interesting high-order functions that I mentioned, the one thing that's really critical about all of them is that you need to frequently update ongoing behavior. If you're writing a paper, you have to frequently update what you're doing, how much you've written. You have to scan the literature. You have to pay attention to what you have done, what you will be doing later. So all of this is really critical. And no matter what you do, whether it's a goal-directed movement or something that is in your mind, the planning stages, updating ongoing behavior live, real-time updating is absolutely critical for cognitive functioning. Now, dopamine should do it, but everybody who records some dopamine cells will tell you, oh, dopamine cells do not encode actions. Dopamine cells encode environmental cues like a tone, and they encode an impending reward. Okay, so I'm going to tell you about the work that Jesse Wood, a graduate student in my lab who just defended his thesis, finished. And Jesse's work actually showed that dopamine neurons encode action number, which is, to me, really important because real-time updates to behavioral strategy requires us to understand how many actions have been executed toward completion of a goal. So to me, this is classic updating of ongoing behavior. Now, how did we show this? He, recording from cells at the VTA, and this is a multi-channel recording, so at the same time he could record from a bunch of cells. And he designed the task, and here's when the old-fashioned task design comes into mind. So he recorded tasks where animals randomly had to do 1 to 20 actions to get a reward. So they always got a reward, but they just didn't know how many actions they had to execute to get the reward. So for example, trial 1, the animal may have had to do 6, and the 7th action was rewarded. Trial 2, the 5th action was rewarded. Trials 179, it was the 9th, and 180 was the 24th. So this randomly changed. So he recorded from 30 minutes. And now, if you actually look at, if you do, if action, just look at the old-fashioned way of looking at activity, which is population activity. What's the phasic response to each action? There's nothing, and that's something that everybody has done. My lab has done. There's really not much of a response to action. But because we had the capacity to record from several neurons simultaneously, what began to emerge is that the same action during the same session, neurons that were recorded at the same time were actually responding to different action numbers. So an example is here. These are 3 neurons in the same animal. And this is their physical response. This is a heat plot, so as the colors get brighter, the neurons are firing faster. So the response is plotted against action number. So you can see that this neuron is sort of more selective toward higher action numbers. This neuron is responding to lower action numbers, 1 to 10, and then this one is much higher action number. So what emerges from the whole data set is this tuning curve of both dopamine and non-dopamine neurons in the VTA, showing that the VTA neurons are diversely tuned to different actions within a given trial. So, and this is actually the tuning curve maxima. So what we see is that the proportion of neurons with 1 or 2 tuning curve maxima are shown here. The action number was modulated about the firing activity of about half of the neurons. And most neurons had a single maximum, and a few other had 2 maxima. But what was interesting is that there is this amazing pattern of tuning curve. We have done a fair amount of computation with this data set. Even if you're interested, I have the entire manuscript and some of the extra data with me, and I'm happy to share them with you. Okay. So summarizing the first set of the data that we have in terms of connecting the fast spiking of dopamine cells to something that's more complex, we think that dopamine neurons encode low, medium, or high action numbers toward achieving a goal. And I think this is really important because it allows for real-time updating of behavioral strategies and real-time encoding of behavior by dopamine neurons, which is an entirely new function that can be assigned to dopamine. Okay. So the second set of data is looking at how dopamine cells firing or bursting in response to changes in the environment can actually lead to sustained activation of dopamine release. So I showed you this data set before, and again, what's really interesting is that dopamine goes up when the animal is engaged in the task, but it stays up after the animal has returned to its home cage. The example I showed you was in a maze, but you also, if you record from dopamine cells, if an animal is doing a task, it could be a cognitive task or a reward-driven task, what you see is that the dopamine cells burst continuously in response to reward, and they continue that for the 20 minutes or so that they are in the operand box or in the maze. So what we decided to do was, okay, let's just take an animal, and instead of making the dopamine cells burst like we do in a task, let's stimulate the cells. First, we started doing it electrically, and then more recently optogenetically, and stimulate them in patterns of bursts for an entire 5 minutes or 20 minutes or 30 minutes, essentially mimicking this pattern of burst activity for just like when animals are in an operant does. So this is essentially what we did. We stimulate dopamine cells using a pattern of activation that resembles phasic bursting of dopamine activity. And at the same time that we were doing this, we measured dopamine release in ventral striatum or in the prefrontal cortex. So what we see is that if you electrically stimulate the VTA cells in that pattern, dopamine levels go up, but they do stay up for at least 40 minutes after stimulation is terminated. And this is essentially the data in nucleus accumbens. So here we are stimulating the prefrontal. These are awake animals, stimulating the VTA, measuring dopamine with microdialysis. This is the period of stimulation. We did both 5-minute stimulation and 20-minute stimulation. Dopamine goes up and stimulation stops. The animal stops being hyperactive, but dopamine levels are sustained for 20-40 minutes. This is nothing like weird or toxic because you can keep stimulating and you see this pattern of activation, very much like when you put the animal in a maze and they do the task just fine. What's interesting is that this happens in the nucleus accumbens and it also happens in the prefrontal cortex. So dopamine in the prefrontal cortex is staying up a good 20-40 minutes after the dopamine cells have come back to baseline. Okay. So this is actually relatively old data. We've been sitting on it for a few years because there are also concerns about with electrical stimulation, you're also driving GABA cells and feedback loops and et cetera. So what brought this data back to life was that we were doing optogenetics and we have TH3 animals, so we could go back and do the exact same pattern of stimulation using optogenetic activation of dopamine cells. And the results are almost identical to electrical stimulation. So here in the dotted line is the data I showed you with electrical stim, and here is with optogenetic stimulation of dopamine cells in the VTA. So dopamine goes up, beautifully responds, but then after we stop stimulation, it's still staying up. Okay. So the main mechanism for this was that, obviously this is expected because you're getting a big burst of dopamine, so dopamine should be floating around. It's not taken up. But what we have found is that it's in fact not spillover, but it is active exocytotic action potential driven release of dopamine. Why? Well, if you put tetrodotoxin in the terminal areas, which should block action potentials, it should block impulse flow, this excess dopamine is blocked, and that's shown here. So if you put, and that's the beauty of in vivo methods, is that you can manipulate a number of different things in an awake animal. So what we see is that if you put tetrodotoxin in locally, the levels come down. So when dopamine cells burst, something else kicks in. A secondary mechanism kicks in that is then enhancing dopamine in the terminal areas. And it's almost like, let's say you really, really, really like a piece of chocolate, and you eat it, you bite, it's awesome, you swallow, and probably dopamine cells burst and yum, yum, but that feeling of, oh, this was great, this tasted really good, that lasts 10, 15, 20 minutes. So this is, it could essentially be something like that. So then the question was, what is the mechanism? And very quickly, we actually have a molecular mechanism for this. I'm going to go through it carefully because I don't see a lot of biochemists or molecular biologists in the field, but we tapped into what was known with amphetamine acting on dopamine transporters and internalizing them. When there's a big bolus of dopamine increase, such as is the case with dopamine, what happens is that dopamine transporters are internalized transiently for about 20 minutes to an hour. So then, and the molecular pathways for that has been worked out, and so we essentially took advantage of this. One of the mechanisms is driven by this Rho GTPase driven mechanism to PKA that's coupled to D1, D5 receptors. So we hypothesized that, maybe this is not just some amphetamine-related mechanism. Maybe this is actually happening in real brain every time our dopamine cells burst. So when they burst, there's a big synaptic increase in dopamine, which then transiently is producing that internalization. And so we actually thought this could be the mechanism. And I'm going to skip this just to show you that this is in fact happening with dopamine. These are in slices. But what we find is that if we actually go and reverse this internalization using a PKA D1, D5 activator, SKF3938, is that this delayed activation of dopamine is blocked. So here we are doing the VTA stimulation. We can do either optogenetic or electrical. And if you block this mechanism, then you don't have that sustained release of dopamine. So essentially, the mechanism that we are proposing is that usually when the dopamine cells are firing in a regular tonic mode, there is dopamine that's being released. And this dopamine, of course, can be taken up by DAT, dopamine transporter. When dopamine cells burst, there is a big release of dopamine. And what happens then is that some of the dopamine transporter is internalized. So because there's less expression of dopamine transporter, a minute or two later, or a few seconds later even, when dopamine cells stop to burst, and they're still releasing the same amount of dopamine, there's far more dopamine in the extracellular fluid and at the surface of the dopamine receptors because dopamine transporters have been internalized. So there is no such thing as tonic phasic in the context of release. These two mechanisms can be very much connected, that one causes the next. Okay. So the second summary is that burst activity of dopamine neurons actually leads to the slow phase of the dopamine cell, which could explain why even the dopamine cells fire so fast. The dopamine does play a role in the slower functions such as goal-directed behavior, motivation, and attention. And so those interesting things are really helping to explain some of the research some of the questions that I've had. So I'm just going to show you a little bit of ongoing data which aren't really clarifying things and making things a little bit more interesting. So we are also, of course, very interested in how driving the dopamine cells. I show you release studies that when we drive dopamine cells, there's prolonged release. We're also interested in what happens postsynaptically in prefrontal cortex and in nucleus accumbens. So one of the experiments that is going on, this is the work of Suhail Orhani, who's a graduate student in the lab, is driving the dopamine cells optogenetically while we are recording units and NLFPs in the prefrontal cortex. And we are using the same patterns of stimulation, of course, different patterns, but also the same patterns that we see happen while animals are doing a cognitive task. So there's a lot of you, if are involved in any psychiatric disorder, prefrontal cortex research, there's a fair amount of literature suggesting that dopamine is related to gamma oscillations in the prefrontal cortex. So one of the first things that we expected was a very nice shift in gamma oscillations. And so far, the data is showing that no effect on low gamma or theta, but a pretty robust effect on high gamma when we drive the dopamine cells optogenetically, and this is in the prefrontal cortex. In terms of unit activity, the effect, so for this is just the results from one animal, just to show that we see a mix of things, both activation and inhibition, or not much. So it would be really interesting to actually phase log some of these basic responses with LFB oscillations in the prefrontal cortex. And another line of research is to look and see how VTA dopamine activity modulates brain-wide activity. This is, again, interesting very much in the context of human work, because with human studies, of course, we're limited to fMRI and PET. So what we are doing is we are combining optogenetics in an fMRI scanner and looking at what happens if we drive dopamine cells to brain-wide activity, and we look at, of course, it's a measure of blood flow. And this is when also very unexpected results have been found so far, because again, we thought if we drive dopamine cells, we're going to see some change in the areas that are receiving input. But results from about 8 animals so far show that, in fact, what the area that's highly activated is the dorsal striatum, which does not even get projections from the VTA. So even though we have all these fantastic theories about dopamine cell firing, what it does to prefrontal cortex, ventral striatum, the regions that could potentially be activated by dopamine cells may not necessarily be regions that are getting direct input from. So how does dopamine do all that? Well, we're still trying to figure it out, and we will work at it, but it's a wonderful time to be doing the sort of work that we do because we have so many wonderful tools at our possession. So I'm just going to stop and to thank the, first and foremost, American taxpayers for supporting most of my research. NIH has been supporting my work for about 26 years. I have a wonderful group of collaborators and an amazing group of people who work in my lab, including Lazio, who we had the pleasure of having him in the lab for about a year. And these are the names of people who have been in the lab recently. And the data I showed you was done by Yomba Kim, Sueda Lohani, Adriel Martigue, and Jesse Wood. So with this, thank you again for inviting me, and it was a pleasure to have the chance to tell you a little bit about the work that goes on in my lab.